Cytokine Release Syndrome, commonly known as cytokine storm, is a severe systemic inflammatory response that can be triggered by various immuno-oncology therapies, including CAR T-cell treatment, infectious diseases, or immune reactions following organ transplantation. Without timely intervention, CRS can rapidly progress to multiple organ failure and death, making it one of the most life-threatening complications in modern immunotherapy. IDC007, developed by IDC, is a groundbreaking bispecific antibody therapeutic that simultaneously targets the major molecular drivers of CRS. By intercepting the fundamental mechanisms that cause immune storms to escalate into severe, life-threatening conditions, IDC007 represents a critical therapeutic breakthrough that could significantly advance the success and safety of immune-based cancer treatments. So how exactly does IDC007 work? Let's dive into the mechanism of action.
CRS is a serious adverse event that can occur during immune cancer therapy, characterized by the explosive release of large quantities of inflammatory cytokines from activated immune cells. During this process, key inflammatory mediators, including TNF-α, interleukin-6, and interferon gamma, are rapidly overproduced. If uncontrolled, this can lead to severe vascular damage and organ dysfunction, creating life-threatening conditions. IDC007 is a bispecific antibody therapeutic that simultaneously blocks TNF-α and interleukin-6, the two primary inflammatory mediators driving CRS, using a single antibody platform. What sets IDC007 apart is its ability to intervene during the critical early phase of CRS development. CRS typically escalate rapidly in its initial stages. IDC007 works by blocking the two cytokine signals that surge earliest and highest during this window, effectively breaking the inflammatory cascade before it can amplify. By intercepting CRS at this crucial early stage, IDC007 has the potential to prevent progression to severe, life-threatening manifestations. Traditional treatment strategies have relied on targeting either TNF-α or interleukin-6 receptor individually, or administering two separate drugs simultaneously to block both pathways. This dual drug approach presents significant limitations, including substantially increased treatment costs and heightened risk of systemic toxicity, particularly hepatotoxicity. In contrast, IDC007 achieves simultaneous dual pathway inhibition with a single antibody, while demonstrating lower hepatic toxicity compared to combination therapy with two separate monoclonal antibodies. In essence, IDC007 maintains therapeutic efficacy while significantly reducing toxicity, providing a safer treatment approach.
IDC007 is a bispecific antibody that simultaneously blocks TNF-α and interleukin-6, the two most critical inflammatory drivers of CRS. To put it simply, think of IDC007 as deploying two firefighters to put out the blaze instead of just one. We observed not only rapid reduction in inflammatory markers, but also effective suppression of vascular damage and organ injury caused by inflammation. Moreover, by integrating dual functionality into a single antibody platform, we've achieved a streamlined manufacturing process while delivering more stable and predictable therapeutic effects in vivo. A single dose can provide sufficient efficacy, significantly reducing patient burden. Importantly, IDC007 monotherapy demonstrates substantially lower hepatocellular toxicity compared to dual drug administration, thereby minimizing the risk of adverse effects. Ultimately, our vision at Prestige Biopharma IDC is to leverage this bispecific antibody platform technology to expand beyond CRS into other refractory severe inflammatory conditions such as sepsis and acute respiratory distress syndrome. We are committed to developing globally competitive therapeutics through our precision dual-targeting antibody platform,